Welcome to today's Becker-Hickel webinar. Before we start and I introduce today's speaker, I want to say a few words to the course of today's webinar, like usual. During the talks, the hand raise function from Zoom will be deactivated. After the talks, there will be a discussion, like in a normal conference, where you please ask your questions. I will activate this hand raise function for it. You can use it by clicking a small button with a hand symbol on it, somewhere in the Zoom window, usually somewhere on the bottom. And yeah, and then I see that you want to ask a question. I will enable that you can switch on your microphone, and if you want, you can also switch on your camera. Would be appreciated, of course. And yeah, that's actually the course of today's webinar. And now it's my pleasure to announce today's speaker. And this is Marina Shirmanova, who is currently a deputy director of the Institute of Experimental Oncology and Biomedical Technologies, Privolzhsky Research Medical University in Nizhny Novgorod in Russia, and the head of laboratory of fluorescence bioimaging in this institute. I hope I pronounced everything correct.
Yes.
Her scientific efforts are focused on the study of cancer from subcellular to a whole tumor level with the use of fluorescence and phosphorescence bioimaging techniques. And the title of her talk is Insights into Tumor Biology with FLIM-PLIM. We are all very excited, Marina, so let me hand over to you and please start.
Yes, thank you, Hoker, for the introduction. Hello, everyone. My name is Marina Shirmanova. I'm working at the Institute of Experimental Oncology and Biomedical Technologies, Privolzhsky Research Medical University, Nizhny Novgorod, Russia. My lab here is doing cancer research. In our studies, we are trying to understand a complex biological behavior of tumors with the use of optical techniques, including fluorescence lifetime imaging and phosphorescence lifetime imaging. And today I will show you some examples how these techniques can help us to do this, how they can be used for cancer studies, and I will explain why do we like these methods. Here's the outline of my talk. I will start with a short introduction into biology of cancer, then I'll describe the instruments we use, and then I will stop on the results with a 4 point there: energy metabolism, oxygenation, apoptosis, and viscosity of plasma membranes, and then some conclusions will come. You know, cancer is a very complex disease. It's very difficult to cure, and cancer cells differ from normal cells in many ways. And all the tumors share some physiological, biological features like uncontrolled abnormal cell proliferation, invasion, the ability to spread through the body from one organ to another, the process of metastasis. To support high growth rate, cancer cells reprogram all the metabolic pathways, including cellular energetics, biosynthesis, redox balance, and also they have the ability to survive with little oxygen. Hypoxia is also characteristic of cancer. Cancer cells evade apoptosis because apoptosis is a natural mechanism of anti-cancer defense. And there are also two more things to say about cancer. To characterize cancer is heterogeneity. Cancer cells within one tumor and different tumors in different patients are very different genetically and phenotypically. And also cancer cells are very plastic, very flexible, which means that they can easily switch between different physiological states and adapt to unfavorable conditions. And collectively, all these things results in poor prognosis for patients and resistance to therapies. That's why we're interested to learn more about these biological features, about these hallmarks of cancer. Why do we choose? Why do we like fluorescence and phosphorescence lifetime imaging? Because, like all the other optical methods, these techniques are non-invasive. It allows us to work with live cells and live tissues in real time to make direct observation of your subject, of your samples, of your objects from the micro to a macroscopic scale, from subcellular to a whole tumor level. And the key feature of fluorescence lifetime measurements is that lifetime, unlike intensity, doesn't depend on concentration of the fluorophore. This is especially important for in vivo studies and for tissue studies, because you can never achieve homogeneous distribution of a fluorophore within a tumor, because tumor itself is very heterogeneous. And also fluorescence lifetime is sensitive to the molecular environment of a fluorophore, and depending on the fluorophore, it can be such parameters as pH or other ion concentration, viscosity, temperature, conformation of fluorophore, binding to proteins. So many parameters can be measured if you use FLIM and have an appropriate sensor. And also fluorescence lifetime is very sensitive. The fluorescence lifetime techniques is very sensitive to endogenous, to autofluorescence of tissue, which originates from metabolic molecules, metabolic cofactors. This is maybe one of my favorite topics today, because this is label-free approach, more close to clinic. This slide shows a basic principle of fluorescence and phosphorescence lifetime imaging. I would skip technical details because there will be another webinar about this quite soon. So just a few words. The time-resolved imaging that I'm talking about is based on time-correlated single photon counting. This means that you detect you record the time of arrival of photons to the detector after laser pulse. You excite your sample with a laser pulse, typically picosecond or femtosecond. The sample emit photons. These photons come to the detector, and the time of their arrival is recorded, and with each next laser pulse, more and more photons are recorded, and then their distribution is built up, shown here. This is a fluorescence decay curve. If you combine TCS-PC FLIM module with laser scanning microscope, you will have microscopic 2D image, like shown here, in which each pixel color-coded and show you fluorescence lifetime. Currently available FLIM-PLIM systems allow you to observe samples with the subcellular and from the subcellular level to a whole body level. As I mentioned, with fluorescence microscopy, laser scanning microscopy, you have subcellular resolution, and this method perfectly works for in vitro cellular studies and in vivo, and even in patients if you have a system like a microscope with a flexible arm. This is already in clinic. If you need to collect signal from the deep tissue layer, you may use a fiber-based luminescence lifetime spectroscopy, which has a fiber inserted into a thin needle, and with this needle you can collect the photons from the tissue depth, both fluorescence and phosphorescence. And with macroscopic FLIM and PLIM, you may scan a field of view up to 2 centimeter diameter, which is really a size of a whole tumor in mice or quite large tissue sample, which is also very helpful for biological studies. What fluorescence we detect? As I said, fluorescence lifetime imaging is very sensitive to detect endogenous autofluorescence from, for example, metabolic cofactors of the hydrogenases, like reduced NADH and oxidized flavins, FAD. This endogenous fluorescence provide us with metabolic information about these cells or tissue. We also can use genetically encoded sensors on the basis of well-known GFP-like proteins. We use such a sensor to detect apoptotic cells. And we also have synthetic dyes, chemical fluorescent probes to measure viscosity of plasma membranes, and chemical phosphorescent probe to measure oxygen. So the first thing I would like to discuss a little bit is energy metabolism. One of the hallmarks of cancer is well-known Warburg effect. According to the classical view on cancer metabolism, cancer cells, unlike normal cells, have increased level of glycolysis, both anaerobic and aerobic. At the same time, mitochondria can be defective, and so the reduced level of oxidative metabolism, oxidative phosphorylation, is often observed in cancer. But later on, I will show you that it is not the absolute, that this is not an exclusive rule for cancer to have an increased glycolysis compared to normal tissues. Additionally, the cancer cells may use other other energetic substrates to obtain energy in the form of ATP, like amino acid glutamine or fatty acid. And cancer metabolism is very heterogeneous and plastic, so cancer cells switch easily between different metabolic pathways to sustain enhanced proliferation, adapt to low oxygen, hypoxia, and nutrient starvation. In cellular metabolism, there are two key molecules that have fluorescence. They are metabolic cofactors, NADH in their reduced state, and FAD, flavin cofactors, in the oxidized states. These molecules have fluorescence in green range of spectra. What they do in the cells? These cofactors work as a electron carrier, first of all, in the electron transport chain of mitochondria. So this is very important molecule and participate in the thousands of biochemical reactions inside cells. NADH is produced during glycolysis, and both cofactors act as electron carrier donor acceptor in the mitochondrial ATC. Fluorescence lifetime of NADH and FAD strongly depends on the state of the molecule. For NADH, it depends on the binding to protein. If NADH is freely diffusing in the cytoplasm, which this state is associated with glycolytic metabolism, fluorescence lifetime is short. It's about 400 picosecond. But when NADH is protein bound to enzyme, and this state is associated with mitochondrial respiration, fluorescence lifetime is longer. It's about 2 nanoseconds. There is also one more form of NADH should I mention. This is phosphorylated NADH bound to protein. This phosphorylated NADH has much longer fluorescence lifetime than non-phosphorylated NADH, about 4.4 nanosecond. But the concentration of NADPH involved mainly in the biosynthesis of other molecules and redox defense. But the concentration of NADPH inside cancer cells typically very low, typically very low and not sufficient to detect by fluorescence lifetime in physiological conditions. In the case of flavin cofactors, FAD, fluorescence lifetimes depends on the conformation of the molecule. This is not only a cofactor, this is a prosthetic group of the enzyme. So if if this cofactor is in proximity to the enzyme, fluorescence is quenched and fluorescence lifetime is shorter, 0.3 nanosecond. In the unquenched state, called also free state of FAD, fluorescence is longer, 2.7 nanosecond. But interpretation, what about this? For an interpretation of the results of this fluorescence and this FLIM data, I would say that an ADH molecule is much more easily to interpret because it is involved mainly in the cellular energetics, unlike FAD, which is involved in the number of other biochemical reactions. So most of our studies concentrate on NADH fluorescence, but not at FAD. It's very tricky to interpret the data of flavin fluorescence. So depending on the metabolic pathways that prevalate in the cells, we can detect the changes in cellular autofluorescence. If glycolysis dominates in the cells, if the rate of glycolysis increases, NADH fluorescence intensity increases because NADH is produced during glycolysis, but fluorescence lifetime of NADH decreases because glycolytic NADH is in free state. Short fluorescence lifetime. If oxidative phosphorylation dominates, fluorescence intensity of flavins, FAD, increases, and fluorescence lifetime of NADH increases because it is involved in the mitochondrial respiration in the protein-bound state. So using this technique, we can assess kind of relative rate of these two metabolic pathways. This is how fluorescence lifetime imaging of NADH works in reality. This is just a screenshot from SPC image software that most of you already probably used. So what we have here? We have the fluorescence intensity image, fluorescence lifetime image of cultured cancer cells. We see kind of granules inside cells. They are mitochondria, just believe me. We have a distribution of fluorescence lifetime across a whole image. And what to pay attention to if you do FLIM of NADH or other fluorophores. First things to pay attention to is the number of photons in the fluorescence for the per decay. If you do B exponential fitting, like for an ADH, it is recommended to have not less than 5,000 photons per decay curve to have correct fitting. The second thing to pay attention to is the goodness of your fit. The blue dot here in the fluorescence decay are your experimental data, and red line here is a fit. So the high square value, the goodness of the fit, is the best value is 1. So if you have 1, your fit is perfect, excellent. Fitting performed, fitting is correct. And next you can look at your at the values, at the fluorescence lifetime parameters you have. What we have here? We have here T1 value, short fluorescence lifetime, short components, free NADH. This value in physiological conditions is almost constant. We also have T2 value. This is long components, protein-bound NADH lifetime. This fluorescence lifetime, this value variable because it depends on enzymes composition and the amount of NADPH with long fluorescence lifetime in cells if it is present. And also we have a contribution, we assess contributions relative amount of pre and protein bound NADH. And from our experience, relative contributions A1 and A2 values are the most informative for FLIM of NADH for detection metabolic activity during different interventions. Currently, different approaches are used to analyze optical imaging data. They can be generally classified into three classes: fluorescence intensity-based approach, fluorescence lifetime-based approach, and mixed approach. Fluorescence intensity-based redox ratio was suggested quite many years ago in 1970s by Britton Chance. It is calculated initially as the fluorescence intensity of flavins divided by fluorescence intensity of NADH, but then different groups use different formulas. But the meaning is that fluorescence intensity-based redox ratio show us a general metabolic activity of cells, their redox state of the cells, because in more oxidative states, fluorescence intensity of FAD increases, fluorescence intensity decreases of NADH decreases, and so you have increased redox ratio. With FLIM based parameters, you have information about T1, T2, A1, A2 values, fluorescence lifetimes of NADH and FAD. Or you can also measure FRET interaction. FRET is first resonance energy transfer between NADH and tryptophan. This approach was recently developed and suggested by the group of Amasi Periasami. Anyway, fluorescence lifetime imaging can show us a shift between different metabolic pathways in which metabolic cofactors are involved. For example, between glycolysis and oxidative phosphorylation. But of course, these cofactors are involved in the number of processes. Some other metabolic pathways can also contribute. This is slightly more complex story, complicated stories that I'm showing now. And mixed approach developed by group of Melissa Scala used kind of combination of redox ratio and fluorescence lifetimes of NADH and FAD. This is an example of FLIM at the macro scale. What we see here, this is imaging in vivo in live mice. We have colorectal cancer growing on the mouse leg. We see the image of a whole tumor here, mean fluorescence lifetime. We see differences between tumor and normal tissue, muscle. We see the ratio of A1 free NADH and A2 bound NADH ratio that also shows us some differences between tumor and normal. And when we construct phosphorescence decay curves, we see that mean fluorescence lifetime in tumor is shorter than in muscle, indicating a more glycolytic metabolism in the tumor. The same we did with the spectroscopic, FLIM spectroscopic system. The system with a needle that is inserted into the tumor, like shown here, also the same tumor, mouse colorectal cancer, and the probe is inside the tissue. We don't have a picture here, only a fluorescence decay, but the information is the same. Shorter mean fluorescence lifetime in the tumor, comparing with muscles, and greater contribution from free NADH associated with more glycolytic metabolism. So these data show us that tumor is more glycolytic than muscle. We also tried to perform metabolic imaging in red brain tumors. We cannot do this in vivo because these tumor cells were inoculated orthotopically into the red brain. So to see the metabolism of this tumor, we need to excise the tissue to take it out of the body. And what we see here, we did this with two types of tumors: glioblastoma, most aggressive brain tumor, and anaplastic astrocytoma. And we found that these tumors has greater contribution from bound NADH4, indicating to a more oxidative metabolism. So a shift to oxidative phosphorylation was observed in anaplastic astrocytoma. For glioblastoma, we unfortunately couldn't see any differences between tumor and normal tissues. So what we see in the samples taken from patients after operation, this was colorectal cancer and normal colon from patients excised during operation. So we did imaging very quickly and put in and stored the samples in specific conditions published in this paper. So we do not recommend this transportation of the tissue samples on air. Just look at in this paper how to do this. So we had tumors of T2 stage, T4 stage, most aggressive, more invasive tumor, and T3 stage, less aggressive. And if you look at this diagram, you see the ratio of A1 to A2, free to bound, and you see here that tumor of the same type but different stages have different metabolism. This was surprising for us to see, but it turns out that the most aggressive T4 tumors had more glycolytic metabolism because they showed greater contribution from free NADH, but while T3 stage tumor had more oxidative metabolism. So you see that the situation with the metabolism of the tumor can be slightly more complex than initially was suggested. These observations for the patient's tumors are consistent with our previous data that we obtained on the tumor of mouse in vivo. We observed monitored cellular metabolism in the mouse tumor in vivo during tumor growth during long periods of time, from the time when the tumor was very small and very large. And we did fluorescence lifetime microscopy, and these images show you fluorescence intensity of NADH overlapped with second harmonic generation signal from collagen fibers, shown in red here, just to assess a tissue structure. But the main result here is that we do not see, I should also mention this, we do not see any differences in fluorescence lifetime during the growth. T1 and T2 values quite the same for different stages, but the relative contribution of free NADH changed here. We see more glycolytic metabolism, a greater contribution from free NADH when the tumors were very small and when they were very large advanced tumors. So this is, in principle, this is consistent with the general concept of the tumor growth, because when the tumor is small, the cells divide very rapidly and need more energy, more macromolecules to build their bodies. So this is why they are glycolytic. They use glycolysis as a main pathway to obtain energy. And when they are large, advanced, hypoxia developed in such tumor, and they just do not have enough oxygen to support oxidative metabolism. This is why they are glycolytic, but this glycolysis is likely anaerobic. Previous studies from different groups also demonstrated that different tumors may have different metabolic states if compared with normal tissues. Some studies show that glycolytic switch is observed in tumors, but there are also studies, for example, the studies of Angelica group, that shows that tumors, cancer cells may have not glycolytic but oxidative metabolism. So it depends on what type of tumors you investigate, you analyze. It depends on what normal tissues in which, what is your normal tissue in which the tumor is located, what the origin of the tissue, what are the metabolic processes inside. So glycolytic switch is often observed in cancer cells, but this is not an exclusive rule. It cannot be, this statement cannot be applied to all the tumor in the world. I should also say about metabolic heterogeneity. Previously, in the studies of animal tumor models, we found that the tumor cells, cancer cells located in different zones of the tumors may have different metabolic state, may have different FLIM parameters of NADH fluorescence. For example, the cells located in the stroma-rich area, cells are shown in green color here and here, and collagen fibers shown in red color. So the cells located among the collagen fibers were more glycolytic than the cells in the more cellular zones. But if we do in vivo studies and look at on the tumor as it is, we cannot be confident at what type of cells we are looking at. So we just suppose that these are cancer cells, but this also can be like, say, macrophages or fibroblasts or something else. So a tumor is heterogeneous, this is a fact. But if we look at the patient samples, the heterogeneity is much higher than in mouse tumors. So just compare tumor and tumor distant colon. A1 to A2 ratio shown here. Each dot here is a measurement made for each cells from the cells, and each column here are the data for one patient. Tumors are shown here and normal column shown here. You see how big is the distribution of the value of A1 and A2 ratio. So how heterogeneous is metabolism for at the cellular level for each sample. So we observed high degree of intratumoral heterogeneity, and we think that such a heterogeneity can be considered maybe one of the mechanisms that underlie resistance of cancer to therapies, because different properties, metabolic differences, cancer cells with different properties can differently response to the treatment. From this part of the talk, we can make the following conclusions. So optical metabolic imaging based on measurements of fluorescence intensity and lifetime of endogenous cofactors NADH and FAD is a non-invasive and highly sensitive label-free approach to interrogate metabolism of cells in a cell culture and in a tumor in tissue. Optical metabolic imaging is now not limited to culture cells and animal models of cancer, but also extends to patients' tumors. And the last conclusion, this method offers a number of opportunities to improve cancer diagnosis, to prognose the cancer development, and for monitoring of treatment efficacy. The next point of the talk is oxygenation. Hypoxia is another hallmark of cancer. Hypoxia is a feature of locally advanced solid tumors resulting from an imbalance between oxygen supply and oxygen consumption. Hypoxia has a few very important biological consequences for the tumors, because it supports genomic instability and heterogeneity. It increases angiogenesis, it increases glycolysis and anaerobic metabolism. Hypoxic tumors are normally more aggressive and have poor prognosis and more resistant to different types of anti-cancer therapy. So hypoxia is a very bad feature. One of the approaches to assess oxygen level in the tissue is to use phosphorescent probe, chemical dye with phosphorescent properties, and phosphorescence lifetime imaging. The principle of this method is that the phosphorescence is emitted from the triplet state of the phosphorescent dye, and oxygen is also in the triplet state. So after collision of molecular oxygen with the phosphorescent dye, quenching of phosphorescence occur, and phosphorescence intensity and phosphorescence lifetime decreases. So with increasing in oxygen concentration, phosphorescence lifetime decreases. The structure of one of such a dye shown here, the phosphorescent dye from Japanese group, Bumei University. Normally typically such phosphorescent dyes contain transient metals responsible for long phosphorescence lifetime and some organic polymers as a carrier. So by introducing these probes into cells or in a tumor, you can measure phosphorescence lifetime and obtain information about oxygen level. We did such measurements using optical fiber-based spectroscopy. We locally injected the dye into the tumor and measured phosphorescence lifetime in the tumor and in the muscle, just to compare their oxygenation. And what we found, shown here, that phosphorescence lifetime in the tumor was significantly longer than in a muscle, which means that oxygen level in the tumor is lower, so tumor is more hypoxic than normal tissues and muscle as expected. And this data about phosphorescence lifetime correlated well with the standard method for hypoxia measurements. We use hypoxia marker pimanidazole to correlate with phosphorescence lifetime. Here, for the first time, we performed phosphorescence lifetime imaging at the macro scale. We used iridium dye, new complex, phosphorescent complex developed by the group from St. Petersburg, Sergei Tuny group. We also injected this dye locally, and here you observe the whole tumor and muscle. A piece of muscle was right here. So you see differences between muscle and tumor. Again, in tumor, phosphorescence lifetime was longer, which means the tumor has a lower level of oxygen. We use this technique to monitor what's going on in the tumor during chemotherapy. We used spectroscopic system again with a needle and treated mouse tumors with irinotecan. Irinotecan is a standard cytotoxic agent to treat colorectal cancer. It is cytotoxic. It acts on directly on the cells, and it was supposed not to affect blood vessels initially, but what we found here, that oxygenation of the tumor decreases during the treatment. So we measured phosphorescence lifetime in the control untreated tumors and in the tumors treated with irinotecan. So treated tumors had statistically longer phosphorescence lifetime. So after the treatment, the oxygen level decreased. So we consider this effect as a new mechanism of action of this drug that hadn't been known before. So conclusion from this part of the talk. Measurements of oxygenation in living cells and tissues are feasible with fiber optic-based phosphorescence spectroscopy and with macroscopic phosphorescence lifetime imaging. Tumors in vivo exhibit hypoxia in contrast to normal tissue muscle, and using phosphorescence lifetime imaging, oxygen level can be monitored in the course of therapy, for example, like chemotherapy, and cytotoxic agent irinotecan induces tumor hypoxia due to anti-angiogenic mechanism of action. The next point of presentation is apoptosis. Apoptosis, evasion of apoptosis, is another hallmark of cancer. So cancer cells, apoptosis is a programmed cell death. It's a very complicated process. This is a natural mechanism of neoplastic defense, because normally the cells with activated oncogenes and with defective DNA eliminated from the body by apoptotic program. But in cancer, apoptosis is suppressed, and this allows tumor to grow, to develop and progress. But it is not completely true that cancer cells absolutely evade apoptosis, because even in cancer cells, apoptosis can be activated in stress conditions like DNA damage, oxidative or metabolic stress, or after anti-cancer therapy. Apoptosis is a multi-step, energy-dependent process in which a number of intracellular parameters, intracellular pathways are involved. But the dynamic of the changes in different biochemical pathways are not fully understood. So, for example, it is not known how activation of caspase-3. Caspase-3 is a key effector caspase in apoptosis, and cellular energetics and ROS production, reactive oxygen species production, correlate with each other, and what is dynamic of the changes in each of these parameters during apoptosis. This is what we try to look at. To detect apoptotic cells, we use genetically based sensor for caspase-3 activity, MKD-AVD-IRFP. This is new genetically encoded sensor developed by Professor Konstantin Lukyanov from Moscow. The structure of the sensor is typical for FRET-based sensor. It consists of two fluorescent proteins, MK2 and IRFP, linked via a caspase-3 recognizable linker DEVD. When caspase-3 inactive in cells and there is no apoptosis, FRET reaction occurs between these two proteins, and fluorescence lifetime of the donor is shorter. Upon apoptotic stimulation, caspase-3 is activated and cleaves the linker, so FRET reaction between these two proteins doesn't go anymore, and fluorescence lifetime becomes longer. This sensor was constructed specifically for live tissue studies because it is red. You know that red light penetrate better into the tissue, so this is the advantage of this particular sensor. And red light can be easily combined with other colors, blue, green, and yellow fluorophores for multiparametric imaging. This is a demonstration. This is an example of in vivo macroscopic FLIM of mouse tumor labeled with a sensor. So on the left, you see the tumor stably expressing the sensor for caspase-3 activity. And as FRET reaction goes between these two proteins, fluorescence lifetime is shorter. It is about 1.8 nanosecond. And this tumor was genetically labeled with a donor alone when no FRET care. And this tumor has longer fluorescence lifetime. But this is just an example. In practice, when you induce apoptosis in a tumor, you can never see such a situation when no FRET occur because the fraction, the population of apoptotic cells is relatively small even after anti-cancer treatment. An important thing here is that fluorescence lifetime imaging and measurements of FAD with this sensor can be done simultaneously with the measurements of fluorescence from NADH. So you can do simultaneously the imaging of caspase-3 activity and NADH. This is what I'm going to show you now. We did these experiments in the cell culture. We correlated caspase-3 activity. We measured fluorescence lifetime of the donor protein in dynamic, shown here, and simultaneously in the same cells, we measured cellular metabolism. We measured fluorescence lifetime of NADH. So cell to cell, the same the same fields of view. So, and what we found here is caspase-3 activity is maximum only after 2 hours after incubation of with staurosporine. Staurosporine is a standard agent to induce apoptosis in the model systems, and metabolism changed very quickly already after 15 minutes after apoptotic stimulation. So we see that the metabolic changes precede the apoptotic cell death. So we see that the fluorescence, a contribution from free NADH decreases with time here, and the redox ratio FAD divided by NADH fluorescence increases, and this data indicate that oxidative metabolism increases before apoptosis. Caspase-3 was activated and apoptosis was active in cells. So metabolic changes are relatively early event associated with induction of apoptosis. Since cellular mitochondria is a major source of reactive oxygen species, it was rational to correlate the observed metabolic changes, increasing the oxidative metabolism, with production of intracellular reactive oxygen species using standard chemical dye, DCFDA. So, and we found that at the cellular single cell level, these two parameters, NADH lifetime and concentration of ROS in the cells, correlate very well. With increase in oxidative metabolism, we see an increase in the production of ROS, which confirm the hypothesis that electron transport chain is a major source of ROS in staurosporine-induced apoptosis. This is live cell. This was shown for the first time. Conclusion 3. Fluorescence lifetime imaging enables to detect caspase-3 activation due to the increase of fluorescence lifetime of the donor protein in FAD-based genetically encoded sensor. The advantage of the redshifted sensor is the possibility to perform imaging in cellular metabolism, simultaneous imaging of cellular metabolism during apoptosis, and activation of mitochondrial oxidative phosphorylation results in ROS production and precedes caspase-3 activation during staurosporine-induced apoptosis. And the final point of my talk is viscosity of plasma membrane. Viscosity is reciprocal of fluidity. You know that the plasma membrane of cells is fluid system, and it determines a number of reactions, a number of properties associated with the transport of molecules to the cells, with the diffusion-related reactions, activity of membrane enzymes, molecular interaction, cell signal, and some other processes. So this is very important to measure viscosity, because viscosity is a parameter responsible for cellular homeostasis. viscosity of the membranes depends on the lipid composition. The presence of cholesterol, the content of cholesterol, which make membrane more viscous, on the length and saturation of fatty acid in the lipids, and on the presence of phospholipid molecules like sphingolipids. In cancer, viscosity of plasma membrane increasing viscosity of plasma membrane is often associated with resistance to drug. And this fact has very simple explanation, because when the viscosity of plasma membrane is high, the diffusion of the drug into the cells is prevented. To visualize viscosity, to measure viscosity of membranes, we used this chemical dye, molecular fluorescent rotor. The principle of such measurements is that upon excitation with light, the rotor, this molecule, rotates, and the rotation rates depends strongly on the viscosity of the solvent. In viscous media, the rotation is slow, and so fluorescence intensity and lifetime of the rotor increases. When you incorporate this molecule into cellular compartments, like cellular membrane or cell cytoplasm, you can measure viscosity and obtain a map of lifetime and viscosity, calculate the viscosity from fluorescence microscopy, lifetime microscopy. We performed such measurements for cancer cells in vitro and compared the viscosity between in vitro cultured cells and in vivo in mouse tumor. What we see here, we see the cells, cultured cancer cells, stained with a molecular rotor. You see here that the sensor is quite specific for plasma membranes. It stains plasma membranes perfectly. You see fluorescence only in the membranes. It looks good. But when you inject the sensor, the rotor into mice, it accumulates everywhere in the tissue. You don't see any structure clearly, but fluorescence lifetime measured in vitro and in vivo quite the same. So we suppose that even in vivo, the sensor accumulate in the plasma membranes, and the value of viscosity is quite the same. This is quite surprising results for us, because the cells were in different conditions. These are the cell culture, these are the tumor, different state of the cells, but the same viscosity. This indicates that the viscosity is quite stable parameter, the parameter of cellular homeostasis, the cellular stability. To ensure that viscosity in the natural and physiological conditions doesn't depend on anything, we measured viscosity in the model of tumor spheroids. Tumor spheroids are multicellular aggregates composed of cells interacting with each other. This is kind of in vitro model of tumor node, and different states, different cells in the spheroids have different metabolic activity and different proliferative capacities. But viscosity didn't differ between different cells. You see here we measured viscosity in the central part of the spheroids, in the cells on the periphery, and it also didn't differ between the spheroids or different size. So we confirmed that viscosity is relatively stable parameter which doesn't depend on metabolic and proliferative activity of cells. But viscosity was affected by cisplatin. Cisplatin is a chemotherapeutic agent, standard agent to treat colorectal cancer. We stained the cultured cells with body P again, with molecular rotor, and treat them with cisplatin. And we see here an increase in fluorescence lifetime and increase in viscosity in 6 hours incubation. This may testify to a new mechanism of action of cisplatin, non-specific, non-related to DNA damages, or maybe just a adaptation of cells to the drug. We don't know it yet, but this is quite bad. Increasing viscosity is quite bad in terms of further diffusion of the drug in the cells. So conclusion 4, for the first time we showed the possibility to visualize viscosity at the microscopic level in cultured cells and in tumor using molecular water and fluorescence lifetime microscopy. Microviscosity of a cell membrane is a stable parameter, and chemotherapy with cisplatin changes membrane viscosity irrespective to the response to the treatment. Summary. Fluorescence and phosphorescence lifetime imaging is a non-invasive, highly sensitive approach, highly sensitive techniques, and allow to, as I show you, they allow to probe different physiological parameters, cellular metabolism, oxygenation, apoptosis, viscosity, and some others if you have an appropriate sensor. Multiparametric approach with a simultaneous probing of several parameters in the same cell or the same tumor provides complementary information about physiology of tumor and response to the therapy. Tumor structure anyway is very, very complex, and biological behavior is complex, so the use of patient-derived tumor material, for example, live tissue slides or primary cell culture or ex vivo tissue samples, can provide more realistic information. I would like to acknowledge my group and people who contributed to this work. I would like to thank our collaborators from clinics and from Baker-Hickel Company, from the Institute of Bioorganic Chemistry, Imperial College London, University of Cork, St. Petersburg University, and Russian foundations for financial support of all these studies. I also should say that there are some unpublished results in my presentation, so the recording of this presentation will not be available on the Baker-Hickel webpage. I'm very sorry about this, but if you need any details about these studies, you're welcome to write me an email shown here. So thank you very much for your attention.